Okay, so this is another 3-point mapping problem, problem 516. Let's see if we can go through this relatively quickly. I went through 514 slowly, so let's see if we can use what we learned in 514 to go through this one at a somewhat quicker pace. So 516 starts off with Drosophila again. So we're looking at a dicate. I'm not sure how to pronounce that crazy phenotype, but dicate is a mutation on chromosome 3 with a dominant effect on wing shape. Okay, so we're going to have D, which is the mutant allele, is dominant to the wild type, D+, and the phenotype is dicate, or dicety. I'm going to say dicate. Okay, D plus is wild type, and then the question tells us in the next sentence that it is lethal when homozygous. So homozygous mutants, lethal. Okay, so we're also looking at two other genes, and these are straightforward. Ebony, we have E+, which is wild type, E+, E, wild type, and then two recessive E's, that's the ebony body color, and pink for pink eye. Okay, this is also a recessive mutation, so P+, is wild type, P+, is wild type, and P, little p, is pink eye. Okay, so we have all the alleles now, and we are going to, the question says, flies from a dicate stock were crossed to homozygous ebony pink flies, and the F1 progeny with a dicate phenotype were back-crossed to the ebony pink homozygotes. Okay, let's map that out here. Okay, so let's map this out. We have to cross flies from a dicate stock. So a dicate stock, one of the first things we should do is assume a gene order. I'm going to assume D is over here. Let's go with ebony in the middle and P on this side. And so flies from a dicate stock, now that could be a little confusing because we haven't talked about fly stocks. But so Drosophila lab will generally have lines of flies of certain genotypes, and with the dicate phenotype, we're going to have, those flies are going to have to be a mixture, because dicate is homozygous lethal. So some of the genotypes are going to be like this. So wild type for E and P, of course, but heterozygous for D, and some of them are going to be just wild type. And people who are maintaining these stocks would just have to go through once in a while and make sure that, you know, we have a good number of dicate flies in there. These are the flies that are going to carry that mutant allele. So the question says, flies from a dicate stock were crossed to, let's see, homozygous ebony pink flies. Okay, so that means these flies, they're ebony pink, so that means they're wild type for the dicate allele, and they're homozygous recessive for the ebony and pink alleles. Okay, so those were crossed, and we'll call this, we're going to call this the P1? Yeah, we'll just call this the P1. So now we have, and it says, the question says, the F1 progeny with a dicate phenotype were back-crossed to the ebony pink homozygotes. Okay, so what does that mean? Let's look at the possible gametes that can be made from this parent. Only one possible gamete, right? Each gamete has a wild type D allele and the mutant E and P alleles. That's the only possibility from that fly. From this one over here, we have D, the mutant version of D, with wild type E and wild type P alleles. And let's see if I can erase this. I'm going to cut that out of there. Perfect. Okay, so this is one type of gamete here, and the other one is going to just have the D plus allele. So two genetically distinct types of gametes from this fly, and only one from over here. Now, the question says that the flies with the dicate phenotype were specifically chosen and backcrossed. So that means we are going to choose those F1 flies that combine this genotype, this gamete, with that gamete, because those are the only flies with the dicate phenotype. So when we combine that gamete with this gamete, so we get this arrangement of alleles in the F1 fly with the dicate phenotype. So it has the dicate phenotype because we selected the gamete with the dicate dominant mutant allele. Okay, so these flies are taken and crossed, back-crossed, it said, to the, let's see, ebony pink homozygotes. So D, E plus, P plus. No, that's not right. D plus, E, P, D plus, E, P. So that was a lot of... Okay, let me see one more thing. Using the results of this back cross shown in the table, diagram this cross showing the genotypes of the parents and offspring of both crosses. And then part B is what is the sequence and interlocus distance between these three genes. Okay, so part A again. Diagram this cross showing the genotypes of the parents and offspring of both crosses, and we've already done that. So look up here, and this was the genotype of the P1 parent from the dicate stock, and we know that because we specifically picked the F1 progeny that had the dicate phenotype. So the only way we could have gotten an F1 progeny with this dicate phenotype was for this P1 parent to have been chosen from the dicate stock. So we crossed this one with the homozygous recessive fly for ebony and pink, and that's the genotype right there. So that's the diagram of the P1 cross. And the F1 cross right here, well, the F1 progeny was back crossed to a homozygous recessive fly for ebony and pink. And that leads to the F2 generation. So that's part A. We've already diagrammed the crosses and the offspring. Now we need to... Okay, so now using the results of the back cross, we need to determine the order of the genes on the chromosome, the order of, or determine which gene is in the middle, right? Dicate, ebony, or pink, and then determine the distances between those genes. So we'd have to look at, so what we have to use is this table, and I'm contemplating if I should copy and paste it in, and I think I will. So I'm going to copy and paste. Okay, so this is the cross, the back cross of the F1 fly with the dicate phenotype. Crossed it with that parent right there, that other parent right there, that other fly, and we got a whole bunch of progeny. So 401 have the dicate phenotype, 389 are ebony and pink, and so on and so forth. So this is another 3-point crossing problem, and we'll take the we'll use the approach that we used in class to solving 3-point problems, and we'll just the first thing we want to do is make a prediction, right? Now let's start with what we've already predicted. So dicate on the left, ebony in the middle, and pink on the right, and that's a little misleading, right? So we can only just determine what's in the middle, and we'll just start by predicting ebony is in the middle. Okay, so we are going to start with ebony in the middle, and I'm going to scroll down a little bit here. Okay, so D, E plus, P plus, D plus, E, P. Now this is the genotype. In the heterozygous parent, remember one of the parents has to be heterozygous for a 3-point cross for all 3 genes of interest. So, okay, let's predict the non-crossover class. Now the non-crossover class results when there's no crossing over during meiosis between the 3 genes of interest. So we have D, E plus, and P plus making up one gamete, D plus, E, and P making up the other gamete. Okay, that's the non-crossover class. Now let's scroll down a little bit, make some room. Next, let's take a look at what happens if we have a crossing over to the right of E. So little p is moving up, P plus is moving down. So D, E plus, P, D plus, E, P plus. Okay, one gamete there, one gamete there. The next one, let's assume that crossing over is happening here. D plus is moving up, D is moving down. So D plus, E plus, P plus for one gamete, and D, E, P for the other gamete. Okay, now this is the single crossover class 2, and this one's going to be single crossover class 1, and then we have one more, right? The double crossover class. Let's use pink. Now, double crossover, we're assuming that this is moving up, this is moving down, and then this one's moving up, and this one's moving down. Or, more simply, that these guys right here are just going to switch places. Okay, so let me draw those out here. D, E, P plus for one gamete, and D plus, E, P for the other gamete. Okay, and that's the double crossover class. So I've gone through the next steps and realized that I needed more room, so I had to rewrite everything a little smaller. And you may have noticed that in the last, in the first iteration of drawing out these guys, I left out that little plus. So I just put that plus in there. Now let's recall that these are all, the female gametes. Let's assume that that dicate fly we used was a female. It doesn't matter. But let's assume these are female gametes. These are all eggs here. Maybe they could be sperm, but let's just to make things easy, let's assume these are eggs. And these are all the possible genotypes because of meiosis in the female and because of crossing over that could happen between these genes. Now they are going to be fertilized with sperm from the other parent, right? And that other parent had this genotype, D plus EP, it's homozygous for D plus EP. So all of the gametes from that other parent are D plus EP. So I'm going to put those right here. D plus EP, D plus EP. And each offspring is going to result from fertilization of a gamete with this genotype over here by the gamete over here. In each case, the gamete over here on the right is going to have the same genotype. And the reason I'm writing these out is because interpreting the phenotypes is a little more complicated with this problem. Interpreting the phenotypes from the genotypes is a little more complicated than we've seen in our previous problems. So what I want to do is just go through these and indicate the mutant phenotypes of the progeny resulting from unifying or combining the gametes on the left with the gametes on the right. Okay, so when we combine these two right here, one of the non-crossover gametes with this gamete on the right, we get a dicate, a fly with a dicate phenotype, and that's it. Because it has wild-type versions of ebony and pink, but it also has the mutant version of dicate, which is dominant. So those flies are dicate. These next flies right here, combine these two, those are going to be ebony and pink. And again, I'm only indicating the mutant phenotypes here. These flies right here, combine this one with this one, and we see they're going to be dicate and pink. These flies right here are going to be ebony only because we have two wild-type dicate alleles. So they're wild-type for that gene, that trait, and we have at least one wild-type P allele, so they're wild-type for that. These right here are going to be wild-type flies because they have two wild-type dicate alleles and they have at least one wild-type ebony and pink. These are going to be triple mutants, right? Dicate because they have one dominant dicate allele and no wild-type ebony and pinks. So dicate, ebony, and pink. These two right here, we have dicate and ebony. Because we have one wild-type dicate allele, the other one's a mutant, and we have one wild-type pink. So it doesn't have the pink mutation, or the pink phenotype. In the last one, the flies are going to be just pink because we have a wild-type ebony and no mutant dicate allele. So next, let us take a look at the phenotypes in the F2 progeny. I'm going to paste this in here, and it's going to cover up some stuff, but I think that's okay. Yes, let's hope it is. So now, so according to the observations of this cross, we have 401 dicate flies. Okay, so we can put 401 up here. We have 389 ebony and pink. Okay, great. 84 dicate and ebony. 84 down here. 96 pink. 2 dicate and pink right here. 3 ebony. 12 dicate ebony and pink. And 13 wild type. Okay, so we can see that we have a problem, right? Because we have higher numbers of progeny that, according to our predictions, should have resulted from a double crossover during meiosis. So at least in the heterozygous parent, and that can't be the case. So the double crossover progeny should have been represented by the smallest numbers of all the progeny in the F2 generation. So let us try to figure out what's going on here. Okay, right? So what do we do here? Well, we need to change our prediction. So what we're going to do is I'm going to get rid of all those here, get rid of all that, get rid of all that, get rid of that. We need to pretty much get rid of everything, right? So we need to make a new model. Let's see if we can do this quickly. So starting over, what we're going to start with is, okay, the previous time we had, what did we predict? E was in the middle? Yeah, E was in the middle. Okay, let's predict P is in the middle. So D plus P, then E. And the genotype of that heterozygous parent is going to look like this. Okay, so let's put down those double crossover. No, let's figure out the different classes of genotypes that result from this order of genes. So let's think here, what are we going for? Okay, just the non-crossover gametes are going to look like this. And I have to remember to write a little smaller this time. Okay, now crossover to the right of P, E is moving down, E plus is moving up. So D plus, P, E plus, and then D, P plus E. And I'm not writing smaller, I should be. I'm going to regret this. Okay, so now D is moving up, D plus is moving down for the other single crossover class. So D, P, E, and then that gives us D plus, P plus, E plus. Okay, now we have the double crossover, crossovers on either side of P. So crossovers over here, crossovers over here, and I'm just going to take the shortcut and I'm just going to move this plus up. D plus, P plus, E over D, P, E plus. Okay. Okay, and that gives us our double crossover class of gametes, our single crossover class 2, single crossover class 1, and non-crossover class. Okay, now these are all going to be fertilized again by plus P, E gametes, only one type. I should say D plus because we're not using the shortcut here. So I'm going to just put those in here. D plus PE, D plus PE, and then we're going to interpret these genotypes again. Let's see how quickly we can do this. Now we're going to interpret the phenotypes. The genotypes are already here. Interpret the phenotypes of the progeny resulting from combining these two types of gametes, the ones on the left with the ones on the right. And what we see is that these are going to be, let me see, I'll use pink here. These are going to be pink and ebony. These are going to be, combining this one with this one, it's going to be dicate. These two right here will give us what? Let's see. pink. These two right here are going to give us dicate, right? Dicate and ebony. These two right here are going to give us dicate, pink, and ebony. These right here are going to give us wild type. These right here are going to give us ebony. And these are going to give us, the last two are going to give us, it looks like pink, dicate and pink. Dicate and pink. Okay, I think these are correct. Okay, so now let's put the numbers in here again. So we go back to the actual data, paste this in here, and let's see if we get the smallest number of progeny in the double crossover class. And we should see the largest number of progeny should be coming from the non-crossover class. So smallest from double crossover, largest from the non-crossover. So choose red now. 401 dicate. Okay, give me my pen. 401 here. Pink and ebony, 389. Pink, 96. Okay, so far so good. Dicate and ebony, 84. Okay, dicate, pink and ebony, 12. Uh-oh. Wild type, 13. Oh, we're okay. Ebony is 3. Dicate and pink is 2. Okay, excellent. Okay, we did it. That was a ton of work. 3-point crossing is a lot of work. We're not quite done yet. So what this tells us is that our prediction of P in the middle is accurate. So the pink gene is on chromosome 3. The question tells us these are all linked on chromosome 3. And P is between D and E. Okay, so we also need to figure out the distance between these. And how do we do that? Well, let's draw our little map down here. Now we have D here, we have P here, E here. Now we just need the number of gametes that resulted from a crossing over between D and P. Now I think everything adds up to 1,000 here. I'm going to assume that these numbers add up to 1,000 of total gametes. And now we just need all the gametes that resulted from a crossover between D and P, and that would be, I'll choose, I can use white now. I haven't used white yet, so white ink. So D and P, let's see, not this one, right? Because the single crossover 1 was the crossover between P and E. So we want this one right here. So this class involved the crossover here, and the double crossovers involved the crossover here. So let's add up all these. 12 plus 13 is 25, plus 3 plus 2 is 5, so 30. So 30 divided by 1,000 total, oh my god, that's pretty small. That's going to be, I think it's going to be equals 3%. So 3 centimorgans. Okay, 3 centimorgans between D and P. Now, what about P and E? Okay, so P and E, so for this distance right here, we need the number of gametes that came from a crossover between P and E. And we can see anything in this class came from a crossover between P and E. So that's 96 plus 84, it's 180. And again, the double crossovers, well, they involve crossovers on both sides of P, so we have to include those in here as well. So 185. So 185 divided by 1,000 is going to be 18.5%, or 18.5 centimorgans. Okay, let me check the answers there. I have to hope we're right. Again, I don't have the hard copy of the study guide. The rep never sent me my hard copy. So I have to log on to the eText version. And I'll pause this for a second. Okay, and I just checked it, and this is the correct answer. Okay, so we did it. A lot of work, these 3-point crossing problems, again, but they help you really, I think, learn a lot about chromosomes and the way alleles cross over during meiosis, and help you learn a lot of really cool things about genetics, I think. Again, come see me if you have any questions on these 3-point crossing problems or any other problem that we talk about in this course.